All right, so a gene is a stretch of DNA or RNA that codes for a protein found at a particular locus or location within a chromosome. So the plural term for that is loci. So you have two loci here containing the gene or the genes, the gene for flower color. Now, an allele, all that is a different gene variant. So the variation would occur in the DNA, so the ATCG sequence. So something within the ATCG sequence would actually be different, making it a variant. We just call that an allele. And so the result of that is going to be a different protein. So if you have a different sequence, you're going to have a different codon. If you have a different codon, sometimes you'll get a different amino acid. And if you have a different amino acid, well, your protein's going to be different. And therefore, the end result is you're going to have a different phenotype, a different physical characteristic. So both of these here are different loci, or sorry, they are the same loci of the same gene, which is for flower color. Now this is an allele for purple flowers, and this is the allele for white flowers. So these are two homologous chromosomes. We're going to assume a diploid, in this case, flowering kind of plant. So this is a diploid here. Here's its homologous pair, meaning that one of these copies, one of these chromosomes came from one parent, and then this chromosome came from another parent. Now, if this was following the rule set of classical Mendelian genetics or simple inheritance, you have an allele for purple flowers, you have an allele for white flowers, assuming, simple inheritance, likely this flower is going to be purple. However, it has two different alleles for that gene, making it a heterozygote, or it is heterozygous. So a mutation, that is just going to be a stable, heritable, new form of a gene, or an allele. I would say that most of our alleles or our gene variants probably emerged from some type of mutation, which can be random. Another term here is wild type. Typically when wild type is mentioned, it's to refer to the most common allele found in nature. So in our examples so far, we've typically, I've only explained or I've only used characteristics with two examples, like purple flowered allele, white flower allele, wrinkled seeds, round seeds. However, for many of our genes, there could be many different potential alleles for a gene. So even though you're a diploid individual, that means you can only carry two alleles, there are many different variants that you could have. So let's say, you could say that alleles are the fruits, and you could only really carry two fruits. So you could carry a banana and an apple, or you can carry a guava and a grape, or you could carry a cherry and a blueberry, for example, or a banana and a banana. So lots of different alleles, but you can only carry two if you're diploid. All right. So in this case, here's an example. We have quite a few phenotypes for rabbits here. We have a dark gray phenotype, a chinchilla fur pattern, a gray fur pattern, a Himalayan fur pattern, and an albino rabbit. Now here it looks like they denote the allele with a C, with the letter C, and then it looks like you have some superscripts, so like lowercase c superscript ch. So in order to be dark gray, for example, you could be big C, little c superscript ch, big C, little c superscript h, big C, little c. So it looks like big C. As long as you've got a big C somewhere in your genotype, you are therefore going to be a dark gray rabbit. Now for chinchillas, it looks like in order to have the chinchilla pattern, you cannot have a big C at all. In fact, the only way to have the chinchilla phenotype or physical characteristic is if you're little c superscript ch. So if you are homozygous little c, ch, or homozygous for this allele, you are therefore chinchilla. Now, in order to have the light gray pattern, it looks like you can have the little c superscript ch, little c superscript h, or you could have little c superscript ch, little c. Meaning you could kind of have a mix, though you must lack big C. So you must lack, you could probably say the dominant allele. And in this case, it looks like you must be a heterozygote containing little c superscript ch in order to be light gray. Now we move on to Himalayan. It looks like little c superscript h is kind of like the determinant for this. So if you are homozygous, little c superscript h, you're Himalayan. Or if you are little c superscript hc, little c, then you could also be Himalayan. So you could be homozygous or you could be heterozygous. And if you're albino, you must be little c, little c. So as you could probably tell from this, not everything is very clear-cut or straightforward when it comes to genetics. It's actually a lot more complicated. There's a lot more alleles that we're not necessarily taking into consideration, especially when I'm explaining it or trying to explain it in a simple way. All right, so this might look familiar, incomplete dominance. So this is actually going to be an example of a type of inheritance where the heterozygote displays an intermediate phenotype. So we have red, which is big R, and then we have white, which is little r. So let's cross the two. We'll cross this R, big R, and this little r, big R, and little r, and what we get are 4 heterozygotes. And actually, I'm going to try to draw this as a Punnett square, because it looks like there's space here. So just because there's space here, I'm going to try doing this. So in order to be red, you are big R, and in order to be white, so this one over here, it could either contribute this gamete or it could contribute this gamete. Because remember, we're all diploid, or at least we're using diploid example. So the gametes are only going to be, they're going to be haploid. So each gamete is going to be contributing one allele, one of the 2 potential alleles. And because these are homozygous, well, they can only really contribute, there's only one potential allele to contribute because they're true bred. So the cross here is big R, little r, which is pink or intermediate. That's the intermediate phenotype. And actually 100% are going to be big R, little r. So with incomplete dominance, you get a intermediate phenotype. Intermediate. So something in between red and white is pink. Right. Now, codominance, this should also look familiar. This is where you have 2 different alleles that are fully expressed. So to kind of piggyback off of the flower example, an example of this would be if you have a heterozygote or a mix of 2 alleles, instead of getting an intermediate phenotype, like the combination of red and white making pink, what you would get is you'd get a flower with splotches of red and white. So just banking off of that previous example, you get a mixture of 2 fully expressed alleles versus, and this is just recapping incomplete dominance, that's where you get that intermediate phenotype, red and white makes pink. So incomplete dominance would be an intermediate phenotype. Codominance is where both alleles are fully expressed. Another example that I typically like to use in my class is blood typing or blood grouping. So if you're type A blood, you're going to only express the type A antigen on the surface of your cells. If you have type B blood, you're only going to express the B type antigen on the surface of your cells. And if you're type O, for example, you're not going to express any antigen on the surface of your cells. And then here's where codominance comes into play. If you are type AB blood, you fully express the A-type antigen and the B-type antigen on the surface of your cells. There's no intermediate, there's no AB hybrid new letter that we're creating what we're doing is we're manifesting both A-type antigen and B-type antigen fully on the surface of our cells. And now we have pleiotropic alleles. So this is where a single allele, or you could say a single gene, is going to lead to more than one phenotypic effect. So in this case, you have one gene leading to multiple effects. So a prime example of this is the fur color pattern, eye color, and crossed eyes of Siamese cats. They're all going to be caused by the same allele. And if those of you who you might not be familiar with Siamese cats, they typically have darker fur or more pigment expressed in the cooler areas. So like their ears, their face, their paws on their front legs and their back legs, and then less pigment expressed in the warmer areas of their body. Now, through a lot of different gene interactions, this kind of downstream of the expression of that gene leads to them kind of developing a cross-eyed kind of look to them, and then also usually a lighter eye color as well. So again, that's where one gene leads to multiple effects. Another example that I typically like to use in my class is natural redheads will also have freckles. So that's an example where one gene will contribute, or one allele will contribute to multiple phenotypes. Now the opposite of that is polygenic inheritance. So this is where you have multiple genes all converging to give you one effect. So a prime example, or there's many prime examples for this. So like height, for example, there's many genes that contribute to height. There's many genes that contribute to hair color. There's many genes that contribute to skin color. I would say that a majority of our genes, or sorry, the majority of our phenotypes, especially the less granular phenotypes, things like cognitive ability or IQ or longevity, those are all going to be examples of polygenic inheritance because multiple genes are going to be contributing to that single effect. And now we have epistasis, and this is where this is a phenomenon where one gene will alter the expression of another gene. Yeah, so one gene is going to alter the expression of another gene. So in this case, it looks like we have three different types of phenotypes. We have agouti, we have black, we have albino. Now, here is an example of a dihybrid cross. We're not necessarily looking at two traits, we're just looking at, you could say, pigment expression or fur color. So in this case, it looks like big A is associated with the agouti fur pattern, and little a is associated with the black fur pattern. And it looks like if you are, or you could say that the agouti pattern, dictated by capital A, is the dominant allele, whereas the little a is the recessive allele. So the black phenotype is actually a recessive trait, meaning you have to be homozygous in order to be fully expressing the black fur pattern. Whereas as long as you have at least one big A, you're therefore going to be agouti. Now, we could study that all day, but simultaneous to this, what's happening, or what is being, what is being conducted, behind the scenes is you're also passing on another gene, or you could say set of alleles, and this set of alleles, so if you are little c, well, it doesn't matter what color you were supposed to be. This big A means you would probably be agouti. Doesn't really matter. Big C, little c, pretty much says you are not going to express pigment altogether. So again, epistasis is just the phenomenon where one gene, so in this case, this little c, these genes, or sorry, this gene, this group of alleles is going to alter the expression of another gene. So that's where you get gene interference. That's epistasis. So now let's talk briefly about sex-linked inheritance. So some alleles are going to be present on the X chromosome, but missing on the Y chromosome. So if we look at this karyotype for a male sexed organism, looks like here we have XY. Now, if you notice here, the X chromosome is much larger than the smaller Y chromosome. And that's because, well, your X chromosome actually carries more genes altogether. And so in the case of, a recessive disease, for example, where you need you need to be a homozygote of a particular gene, or a particular allele. You need to be a homozygote of a particular allele to express the disease. Well, if you're a male and that disease is, carried on, on a loci that the Y doesn't even have, meaning Y is not even carrying the gene, well, you don't need to be homozygous to express that phenotype. You don't need to be homozygous to express the disease. All you need is to have a single allele. And because you have a smaller chromosome here, which doesn't happen to have, you could say, like a buffer for the disease, therefore you're going to be affected by the disease. So some examples of this are going to be like balding, hemophilia, color blindness. And so here we have an example of a male which has the XY, or he's going to be XY, and it looks like blue is an unaffected allele. So this is going to, we'll just use color blindness, for example. So he has, he's unaffected by color blindness, and he has children with a mother who has an affected allele. So she has the recessive allele that if she had both of them, if she had two recessive alleles, she'd be colorblind too. But because she has that extra X chromosome, she's not going to be affected by color blindness. She's not colorblind at all. In fact, she's just a carrier. And so let's say they have some children. Well, we've got a 50-50 chance of having male versus female, right? So this is going to be your XY. This is going to be your XY. Now, if a male receives the recessive allele, that would normally cause disease in a female if it was homozygous. Well, if this male receives a single one of those alleles, he therefore will manifest color blindness. So because he is XY, this Y provides no buffer to the disease. It simply doesn't carry that gene. Now, if the mom passes on her affected allele, or her, so her X chromosome containing that recessive allele, and then it's meshed with, the normal X chromosome from dad, or the normal allele from dad, well, what happens is she has two X chromosomes. One of them acts as a buffer to the disease. Therefore, she's going to be unaffected, but she's going to be a carrier. So therefore, for all of our, or for a lot of our, what we consider to be sex-linked genes, well, it's sex-linked in a way that the X chromosome carries the allele, where the Y chromosome will not carry the allele. Therefore, males are going to be more commonly affected by those diseases. So things like hemophilia, or even like, male pattern baldness is going to be more higher, expressed at a higher rate in males than females. Because males can't be carriers, they can only be affected by the disease. They're either affected or not affected. All right, so last slide here, we got hybrid vigor, or otherwise known as heterosis. And that's just the tendency of a crossbred individual to show, you could say, stronger traits than both parents combined. So qualities that are superior to both parents. So here we have, one parent which has, a long head of corn, but is not as richly dispersed with kernels. So it's a long head of corn that has sparsely dispersed kernels. And we have another parent which is kind of like a short head. It's got a short head, but very richly dispersed kernels. Now, both of these parents mate with each other, and they create offspring, and this offspring is heterotic because it has traits that are enhanced as a result of mixing the genetic contributions of both of these parents. So this hybrid offspring has a long head of corn and is richly dispersed with kernels. So that's an example of hybrid vigor.